Hey guys, so I know a lot of you might be out today because of the pep rally and various other things that are going on, and so I wanted to get on and make sure you didn't miss out on some of the notes. I'm going to put myself here over these plants. So most groups made it through this slide, but in case your class didn't get this far, remember the big difference between autotrophs and heterotrophs is that autotrophs can feed themselves, auto means self, and then heterotrophs have to eat other organisms or get other nutrients in a different way because hetero means other. As we're going through this, if you need to pause to write things down on your note sheet, you're welcome to do that, or you can also view the slides that are in Google Classroom. And there were some examples here. Of course, your plant is an example of an autotroph, and then you have a absorptive heterotroph with the fungi, and then you have a hyena here eating a little baby wildebeest. Sorry if we can hide that. That would be obviously a cannibal, or I'm sorry, carnivore. Moving on to cell size. So this is really where most groups would be picking up today. The big idea with cells is it's really important to be small. And the reason you want to be small is because when you're small, you have a better surface to volume ratio. And so as our cell gets bigger and bigger and bigger, you'll see down here by doing a little bit of simple math, if you had a 1 by 1 by 1 cube, for example, your surface area is 6 and your volume is 1. Whereas if you expand that to a 10 by 10 by 10 cell, you get a 600 to 1000 ratio, which means you're actually at a 0.6. And so the bigger we are, the harder it is for us to bring in what we need and also get out what we need to remove from the cell, whether through endocytosis or exocytosis. And so cells, instead of growing really, really large, will typically divide. A couple other things here on why cell size is typically small. One is it's, as we said, a distance problem. It's easier for us to carry out things like diffusion and active transport if we have a smaller distance. And then also, there's only a certain amount of information that we can contain in one cell. And so if our cell gets too big, then we can't carry out proper metabolic processes, and we would have to divide. Otherwise, we would risk our cell not getting all the nutrients it needs in all the different parts of the cell. There's a short video here, which I would encourage you to go and watch, but we're going to skip it for the sake of this kind of overview section. Let me scoop myself over. And so when we get into section 5 here, it really starts talking about the components on the cell. And what we're going to do is actually go through that chart kind of as a group. And so if you have that chart, you're welcome to follow along and kind of plug some things in. First thing here, of course, is the plasma membrane. And that's what surrounds the cell and separates it from the exterior and the interior of the cellular environment. And so one of the most important things that the cell membrane does is it regulates what can go into out of the cell. Kind of think of it as like the bouncer at the club that looks you up and down and says, oh yeah, you can come in, or no, you can't come in, depending on if we need to or not. Obviously, the channel proteins and stuff, we'll get to that a little bit later, but just on the basic structure, we have our hydrophobic and hydrophilic regions. So remember, the hydrophilic is this head here, which is the part that loves water. So that's going to face the extracellular and intracellular environment. And then the region between those two is hydrophobic. So you can think of that phobia as like being scared of the water. And so it forms an impermeable membrane in the middle of the cell. And so water can't very easily diffuse across this membrane. And if you remember from biology, the diffusion of water, remember, is osmosis. But primarily when we're dealing with the cell, we'll talk about diffusion or active transport. Two more scientists here you need to know about, Singer and Nicholson. They're the ones that came up with the idea of the fluid mosaic model, which is basically that we have this phospholipid bilayer, bi meaning two, and then phospholipid because it's a fatty acid. And we see things that are just embedded within that. For example, what's on the slide here. And so I'll have people actually sketch this on their notes, and I would encourage you to do that. Either this model, or you can do this little basic model right here. And then we have a picture here. This is a Singer sewing machine, and then Jack Nicholson. So Singer and Nicholson, not obviously those people. And a little bit more here. We already kind of mentioned some of these things, so I'll scoop myself out of frame for a second. But you can see we've got the phospholipid bilayer. When anything is labeled as water soluble, that simply means that it, if you read here, is relatively impermeable to water soluble molecules. In other words, water and other things that are dissolved in the water can't go across the membrane unless the cell wants it to, specifically with channel proteins or other carrier molecules. We've defined here hydrophobic and hydrophilic, water-fearing and loving. And then the proteins, which not only let things across the cell, but also, if you remember from the reading quiz, they're really important for cell recognition. They're markers for that and also hormone receptors. And then lastly, there's two types of protein. We have the integral and then we have intrinsic. Those are sometimes used kind of interchangeably, but basically those are proteins that are inserted in the bilayer. So if you have your bilayer here, I realize I'm doing things and I'm off screen. So if you have your bilayer here, a protein would be something that cuts across that and allows for the movement back and forth across the membrane. And typically that's for like ions, whether hydrogen ions or something else that needs to get into the cell. There's this chart, which is in the notes, that we actually are going to kind of skip because we don't really actually need to know as much. But if you're curious what the proteins look like, you can see that. This is some of the examples. We have things like channel proteins, we have receptor proteins, we have enzymes, which you might remember from biology help carry out those reactions. We have binding proteins, which allow cells to bind with other cells. Some cells will actually do this, not human cells, but bacteria cells, in order to do conjugation, aka the transfer of genetic information between cells, which is kind of a neat deal. And then there's also things like filaments and markers that can be found on proteins, and that's what helps us figure out things like what was on the reading quiz about whether it's a good or a bad cell, self versus a non-self. Okay, going a little further, the peripheral or extrinsic, that means it's attached to the membrane surface. So extrinsic, kind of think like exterior of the cell. And then things like cholesterol is really important to the cell membrane. Cholesterol, remember, is a lipid, and that's what allows the membrane to be more fluid. And so if we didn't have that cholesterol, if you don't have proper cholesterol levels, your cell membranes actually become very stiff, and it can damage your cells. And so it's really important to have a proper amount of cholesterol. A couple of specializations, and these are ones that were on the reading quiz. Your microvilli, you need to know that, and they're pictured here. Those are your tiny kind of finger-like projections, and they kind of stick up on off of the cell surface and kind of wave like this if you do your little jazz fingers. And those are helpful for absorbing material into the cell. Typically, you'll find this on like the kidney tubules or your small intestine, where your stomach is digesting and then passing that material on to be absorbed into your body. But you can also see it in some other regions as well. Membrane junctions, that's just talking about the various different glycoproteins that are on the surface. They're kind of wavy looking, and there's lots of different types. And we're going to look at a couple different junctions, and then I think that'll be almost as far as we make it today. So let's kind of get these junctions here. Let me scoot down for a minute. So you have really three flavors of junction. You got your tight junctions, your desmosomes, and then your gap junctions. So we'll kind of go through them one at a time here. So tight junctions are really, like they sound, tight. They're kind of like a weld almost, where a seal is formed between two impermeable barriers. And that's helpful because that allows us to not leak fluid, which would be really bad. So we don't want to leak like our digestive juices out of our cells. So for example, in the digestive epithelia, which is the lining of your digestive tract, you'll see things like tight junctions. Desmosomes, if I move up, are spots that weld adjoining cells together, and that's going to help us not basically break apart. For example, like the skin tissue layers are held together through desmosomes, because you don't want part of your skin to fluff off and then like lose all of it. And so you'll have these kind of welded joints between each of the layers of your skin. And then lastly, we have gap junctions, which are perforated channels. In other words, they have little holes. And that allows things to really easily go into and out of the cells in this region. So for example, if we need like electrical current or ions or small molecules to pass back and forth easily, like in cardiac or in smooth muscle, where we need to move energy basically and be accessible, we're going to have gap junctions that join those different regions. And so there's a really handy picture here. You can see the different types. So we have our desmosome, where you can see that weld. We can see a gap junction here, which has those perforations. And then we have a tight junction where it has kind of those similar to the desmosome with a weld. But remember that a tight junction is really there for fluid leakage sealing. And so you can see how things could be flowing, but not kind of crossing that barrier. And you can see this is actually skin right up here. Okay, and then lastly, with plant cells instead of animal cells, the plasmodesmata are bridges of cytoplasm, which you can actually see in the image here, kind of this plasmodesma, which is this kind of linkage between adjacent cells. And what they do is kind of form these bridges where you can go from one cell on a plant to another cell. And so that's kind of handy for cells in plants, at least, to be able to transport molecules. This is kind of a microscopic view of it. It's a little bit harder to see the actual plasmodesma in this, but in the illustration you can see it really, really well. And this is not in your textbook, so that's why we're kind of showing some different diagrams. Okay, lastly, let's think about membrane transport. So with membrane transport, we'll see that primarily it's going to occur through a very passive process. And that means it doesn't require any energy. You just get on one side and things are going to carry you to the other side because of diffusion, where it's just down a concentration gradient. Similar to when you get on the bus, you don't have to do anything. You just sit there, or the train, and it just takes you where you need to go. Whereas on the other hand, something like active transport, go forward, is going to require ATP. There's an example here with a lump of sugar. So if we drop a sugar cube in a beaker of water, those molecules are going to spread out because we go from a really high concentration of sugar, and then it's going to spread out over time so that we have an equal distribution of sugar molecules within the water. This diagram is showing a solution where we have 10% glucose and 20% glucose. And if we wait long enough and there's a permeable membrane that's both permeable to water and glucose, then we'll see that they balance. We're going to end up with 15 to 15%. Compared to if we do something like osmosis here, where we can only allow the water to move through the membrane, then in order to achieve equilibrium, we might actually see different levels of fluid on one side or the other. So osmosis, you'll notice the diffusion of water, whereas diffusion is more the movement of particles. And that might be water, but it might also be like ions and other things. Facilitated diffusion is very simply just diffusion that needs help. So for example, diffusion that takes place through a membrane channel, or you can see this one is closed, so it's not able to go through. So instead of being just basic diffusion where it goes across on its own, it just needs a little help in order to achieve its goals. So for example, like glucose gets into the cells like this through a carrier protein. There's a neat little GIF here, but it seems to not be working right now, which shows those molecules going across. Oh, there it goes. Eee. Okay, don't clip that. Lastly, we talked about osmosis, but at the bottom here is osmolarity. You might remember molarity from chemistry, but you should know that osmolarity is where we have a total concentration of all solute particles in a solution. So by looking at this percentage here of albumin, we're actually looking at the osmolarity of these solutions. And you'll see that water moves to reach equilibrium in osmolarity. Okay, and then the last biology review, I think, is tonicity. You'll remember tonicity is, there's really three types. There's isotonic, hypotonic, and hypertonic. Isotonic means we're kind of equal. Think about like an isosceles triangle. I think that's the one that has all the same sides, kind of equal, equilateral. Hypotonic, hypo means less. So like if you're in a hypotonic solution, you can see that that means there's less concentration outside than inside the cell. And so water is going to want to rush into the cell to balance that out. And so our cell will actually swell up like a balloon. And you can see that here. And it's possible that your cell could even explode, which would be bad. And then we have hypertonic, where the solution outside has more concentration than inside the cell. And so water is going to leave the cell, and your cell is going to shrivel up. And that would also be bad. By the way, I don't think this is on the reading quiz or on anything coming up, but just for funsies, when your cell explodes, it's typically called lysis. So it might be a good thing to know. Okay, let's do, I don't know that we'll make it this far in class, but I'll talk about it just in case. With filtration, your cell membrane is also able to filter and let certain things in, certain things out. And that's typically through a high to low pressure gradient. I put a picture here of a coffee filter because that's kind of how your cells work. When things are forced through the membrane, it kind of filters out certain things, like keeps your coffee behind, but it lets that coffee water pass through. And a lot of this is due to things like active transport, where the cell is actually using energy with carrier proteins to move things across the membrane. And so the classic biology example is the sodium-potassium pump, which is on this next slide. So we can see that there's sodium buildup within the cell, and we need to get it out of the cell while bringing in potassium, that's K. And so in order to do that, our cell has to use energy. And so we have this little place where the ATP molecule can come and bind. You see this little cone of ATP. And as it does that, it's going to change the shape of the carrier protein. So you can see how these go away, that's sodium. And they go outside of the cell. And then the potassium is brought into the cell against the concentration gradient. And so it would make sense for like one of these to maybe go across, but for both of them to come in, that's why it requires energy. Okay, and then we'll end with a little active transport video, and we'll talk about defining these terms. So bulk transport, that just means we're moving a lot of stuff. And I have some pictures here which maybe will make sense if I explain them. We have exocytosis, where we're sending something out of the cell. Endocytosis, we're bringing it into the cell. Endo, inside, exo, outside. We have phagocytosis, where we're eating. That's why there's a hot dog. Penocytosis, where we're drinking. So that's when the cell is bringing in liquid. That's why there's coffee. And then receptor-mediated endocytosis, which is the funnest one to say. But that's basically where there's little proteins on the surface, and things will come and touch those proteins. And when that happens, the cell's going to be like, ooh, yum. And then it's going to wrap itself around that molecule and bring it into the cell. And so that's kind of the mainly two types of bulk transport you would need to know. And here's a couple diagrams of that. So we have phagocytosis, again, where I'm cell eating. Penocytosis, it's called drinking because these are dissolved ions, not a large food or a large particle that's being brought in. And then your receptor-mediated endocytosis, where something is binding to a protein, and that's activating this process. And of course, these all require energy. Okay, we'll stop there.